Okay, now. You know all about this. Litmus cream, right? What do you want? You are so annoying. Here's the litmus cream, okay? You know all about this now. Starts out this color, right? Bluish purple. You take litmus cream and lipase, it doesn't really work, right? See how it just gets a little lighter purple, doesn't really work? When you take litmus cream plus lipase plus bile salts, it works great. See, it turns pink, right? Because in lab 2, I didn't tell you that there's special stuff in the lipase reaction, so it'll work. Because I didn't care about it then. All they wanted you to do is learn what an enzyme is and what it does, right? Lipase digests fats. Now that you're so sophisticated and well educated, now you're able to add an extra detail, which is very, very important, which is that of the bile salts. Okay, now bile is what you get when you break down red blood cells. You take these chunks of lipids from the all red blood cells, right? And we use them to emulsify the fat. In other words, we take the lipids, that are these big, non-polar, hydrophobic molecules, and we break them up into smaller and smaller structures, right? Smaller and smaller droplets. And why do we do that? It's an attempt to make them more soluble, right? Because most enzymes work by hydrolysis, right? They add water across bonds to break things up. Correct? Standard catabolic reaction, right? Now, if you have a lipid, well, it doesn't like to go into water, does it? You have to make it go in, right? So you need an adapter. So the bile salts are an adapter. It's called amphipathic molecules. Means that it has a polar end and a non-polar end. So you can never get away from chemistry, no matter what you do, Rose, see? Never. So I'm saying, just get over it and accept it. Because you can't ever get away from chemistry. You just can't, never. Okay? So what happens, so now you have a non-polar end, right? That's almost like a lipid. And you have a polar end that's like an amino acid or a carbohydrate, right? And it works like an adapter. So the non-polar end goes with the non-polar end of the lipid, and the polar end goes with the water and the enzyme, right? It's insoluble in the water, right? So now the lipid can be found by the lipase and cut it up. And when you cut it up, what do you get? Fatty acids and glycerol, right? And that's why it turns pink, right? Because it's fatty acid. Now, everyone understand how that works? You don't understand how that works, Raquel? Okay. Give me a non-biological example of how that would work, if you understand it. Very simple. Okay? I'll give you a hint. When you wash your clothes, right? Do you wash your clothes or you have somebody else do it for you?
My mom does it.
Oh, cool, okay. Does anyone wash their own clothes in here? Okay, right. Not the princesses, I'm sure, these three. Okay, so now, right? Okay, so now, right? 1, 2, 3, 4 princesses, but the other ones probably do, right? Isabella, you don't go. You don't even know what a dishwasher looks like. A washing machine.
I do my own laundry.
You do?
Yeah.
Good girl. Okay. I said you were one of the princesses too. Anyway, so now it's called edit button. Now. No. Okay. So now what happens is, example of this is you take laundry detergent, the Tide, with whatever you use, right? And you look at it, it's kind of this not really aqueous solution, right? It's got like lipid components to it because it's a phospholipid. It's amphipathic. So when you take it, like shout is the best example. Like stain removers, they're sticky, right? You glob them on and they actually stick to the stain, right? What they're doing is they're lifting the dirt out of the fabric, right? Because what is most dirt? It's lipids, right? Oil, grease, wax, they're all lipids. Okay, so what you do is you put that, spray this stuff on there, right? And you wait. Then when you go to wash it, what happens? It gets thrown around in there, spun around, right? The water and the soap grab the dirt, pull it away from the clothes, and what happens? And it spins at really high speed. Like the new washing machines are really cool. They spin at really high rate, like 2,000 RPM. It's really cool. It's like a little centrifuge. It's so much fun. Okay? You spin it on the highest possible speed, and it spins really fast, so it pulls all the soap away from the clothes. The soap is holding all the dirt, right? Goes down the drain, you rinse it out, you take it out, you get clean clothes, right? Well, your digestive system, you got the idea from your digestive system. That's what your digestive system does. Takes the lipids, chops them up into smaller groups, smaller lipid drops, digests them, turns them into monomers, and sends them into the blood, right? So that's the lipids. Starch. You know this one too. That's the amylase, right? Mm-hmm. Amylase is secreted from the pancreas. It goes to the duct system, through the pancreatic duct to the duodenum. It's mixed with the carbohydrates. It chops them up. It keeps chopping them until you get to monomers. The most common monomer of carbohydrates is glucose, right? Okay. Glucose is going to get absorbed, put into blood, and used to make ATP, right? By the organs. Pepsin. It's an example of a protease, okay? Pepsin is a protease. It will take large proteins in the stomach and chop them up into little peptides. That's its job. So it takes really big proteins, chops them up into little peptides, so they can be finished their digestion in the small intestine. Okay? So pepsin is a protease, okay? So it has to work at an acidic pH, okay? Okay. Look at this one. In normal water, it doesn't work. In neutral pH, it doesn't really work, okay? When you put in acidic pH, and it works great, see? Chops it up. What's the pH of the stomach? Like 2, right? So, the enzymes in the stomach have to be able to work at really low pHs. That one, I don't know why this other one, basic one, it must be gold or something. It has to be replaced. Okay? So the basic one should not have worked. Only the acidic one should work. Okay? So now, the pH of the stomach is around 2. And the pH of the small intestine is like 8. Right? Why is it so drastically different? So the digestive system can compartmentalize, can control what happens and where, right? Okay, you don't want digestive enzymes that have their highest activity at body pHs, right? That would be bad, right? So if you had pepsin worked right at 7.4, that would be really bad, right? Because then if they ever got out, it would start digesting us. This is not good, right? So you want it to be like 8.2 or lower, okay? You want it to be lower than 7 or higher than 8, right? And that's most of those enzymes, that's how they work. So they work only in that compartment only. Because remember, there's no difference between me, biochemically, there's no difference between me and the chicken I eat. You know what I'm saying? Same exact stuff. Right? Okay? And there's no difference biochemically. So, so my enzymes could just as easily digest me than the food I eat. Okay? Because that's why, so you have to be orderly and compartmentalized and safe. That's why pancreatitis is so dangerous. It'll kill someone in a couple hours. Okay? Okay, so that's the thing. So, okay, so look at that stuff. Okay, last thing here is